Hello, dear students. Today, the topic that we are going to discuss is general characters and classification of algae. The lecture comprises of 3 parts. Number 1, introduction to the topic. Second, we will discuss the general characters of algae. And lastly, the classification of algae. So starting with introduction. Algae is a diverse group of living organisms, mostly autotrophic eukaryotes, presently classified under the kingdom Protista. The term algae has been derived from a Latin word algae, meaning seawards. The scientific discipline that deals with the study of algae is known as algology, or very often phycology. The term phycology is derived from 2 Greek words, phykos meaning seaweeds and logos meaning study. Algae are known to occur practically in every habitable environment. However, they are predominantly abundant in aquatic habitats. The occurrence of algae in habitats such as hot springs, ice-damped soil, rocks, tree trunks, old walls, etc. has been also reported. A number of algae can be found as endophytes, epiphytes, and even mutualists. The free-floating assemblage of algae is collectively known as phytoplankton. The phytoplankton is a major contributor to the global primary productivity. However, under eutrophic conditions, such planktonic algae increase their number and extent enormously and form water plumes. Now the second part, general characters of the algae. Algae fundamentally differ from all other autotrophic eukaryotes, that is higher plants, in their production of gametes or gametangia without a covering of sterile cells. The plant body that we call as thallus lacks the distinction into true root and shoot systems. Also, the algae lack true vascular system. Now about its plant body, which we commonly call as thallus. The term thallus has been derived from an undifferentiated vegetative body. The thallus in algae varies from the relative simplicity of a single cell to the most striking complexity as shown by the giant kelps. While as unicellular algae, such as chlorella, with a size of 2 to 8 micrometers, or in the range of bacterial cell, the kelps may attain a length of 60 meters. The algal plant body can occur in various forms. It ranges from unicellular, colonial, filamentous, foliose, and tubular forms to the more highly differentiated blade-like forms and those that have distinction into root-like, stem-like, and leaf-like organs. Unicellular algae form 2 types of colonies. In one form, the number of cells is indefinite. In the second form, the number of cells is definite, and we call them as cenobium. Now discussing the reproduction in algae. Algae reproduce both by asexual and sexual means. However, in some algae, sexual reproduction does not occur at all. It is either through its phylogenetic loss or it has not developed. Asexual reproduction. It involves the increase of progeny without involving the fusion of cells and nuclei. Many algae reproduce by simple fragmentation, in which the fragments have the capacity to increase into new individuals. All the vegetative or somatic plant body which is involved in fragmentation is sometimes called as vegetative reproduction. In addition to fragmentation, algae produce a variety of spores. These spores are produced within an ordinary vegetative cell or within specialized cells. The specialized spore producing structures are known as sporangia. Amongst the various types of spores found in algae, akinetons are of widespread occurrence, especially in the green algae. An akinete is a vegetative algal cell that has enlarged and formed an envelope external to its wall. In many non-motile algae, flagellate zoospores are the agents of asexual reproduction. In some cases, the zoospores bypass their motile phase and start their development within the parental cell wall and are called as aplanospores. Such aplanospores that thicken their walls are known as hypnospores. Other types of non-motile spores produced by algae include autospores, as in green algae, monospores, tetraspores, paraspores, and corpospores, as found in red algae, and statospores and oxospores in diatoms. Now, sexual reproduction. It involves the union of cells, what is known as plasmogamy, the union of their nuclei, karyogamy, and the association of their chromosomes and genes, followed by the meiosis. In sexual reproduction, there is a fusion of gametes which form a zygote. The zygote either directly germinates into a new individual or divides to form a number of zoospores which after liberation germinate into new individuals. Now, depending upon the structure and function of the gametes, three types of sexual reproduction are recognized in algae. Number first, isogamy. It involves the fusion of motile or non-motile gametes which are morphologically and physiologically similar. Anisogamy. It involves the fusion of motile or non-motile gametes which are morphologically dissimilar. Usually, the larger gamete is the female and the smaller one is the male. Third, oogamy. It involves the fusion of a large non-motile gamete called as egg or ovum with a smaller motile gamete called as sperm. Usually, the eggs are formed within an oogane gonium and the sperm within an antheridium. Now the third part of the lecture, that is the classification of the algae. The classification of living organisms has occupied human attention since the time of Aristotle. The traditional system of two kingdom systems of classification of living organisms into plants and animals is no more valid. At present, the living organisms are divided into three main domains. These domains are eubacteria, archaebacteria, and eukarya. Thus, domain and not the kingdom is highest level of taxonomic category. And the domain eukarya, the four kingdoms recognized are Protista, fungi, Animalia, and Plantae. The traditional group of autotrophic living organisms grouped under the algae is now classified under the kingdom Protista. Previously misclassified algal phyla of blue-green algae, commonly known as cyanobacteria, is now classified under the domain eubacteria in the kingdom Monera. In fact, it was Linnaeus, a Swedish naturalist, who in 1753 was the first person who used the term algae for a particular group of plants. However, his group actually consisted of a mixture of algae and liverworts. However, in 1838, for the first time, classified algae into four main groups based on the variations in color of their thallus. These four groups were green algae, red algae, blue algae, and diatoms. Since then, from time to time, several classifications of algae have been proposed based on a variety of characters, including micromorphological, physiological, biochemical, and more recently the molecular ones. Before we start the classification, it is very important that we discuss the main characters that have been used in the classification of algae. Unlike other groups, such as higher plants, the morphology of mature vegetative stages in algae does not allow a reliable criterion for distinguishing different phyla of algae. Some of the main characters that have been widely used for the classification of algae are photosynthetic pigments, second, nature of food reserve, cell wall composition, and flagella. Photosynthetic pigments. In algae, three main types of photosynthetic pigments are known: chlorophylls, carotenoids, and phycobilins. First, the chlorophylls. The photosynthetic algae possess chlorophylls in their chloroplasts. 5 kinds of chlorophylls, A, B, C, D, and E, have been reported from different groups of algae. Carotenoids. These are groups of protective pigments that pass on the light energy to chlorophylls after screening. They occur usually inside plastids. Generally, the carotenoids can be divided into two classes. One, carotenes, second, xanthophylls. Phycobilins. It is basically a pigment protein complex. These are tetrapyrrolic compounds joined to globulin proteins. These are water-soluble blue or red pigments located on the thallocytes or inside the thallocytes of chloroplasts. Based on their absorption spectra, 3 types of phycobilins recognized are phycocyanin, phycoerythrin, and allophycocyanin. Second criterion was the nature of the food reserve. The primary product of photosynthesis in all the algal phyla is same, that is glucose. However, the insoluble food reserve products which accumulate over a period of time are variable and thus provide useful characters for classification. Polysaccharides, in which glucose subunits are linked together by alpha-1,4 linkage, or starch and Floridian starch, and those with beta-1,3 linkage or laminarin, chrysolaminarin, and paramylon. Third criterion was the cell wall composition. The chemical composition of cell wall varies among various algal phyla. In general, algal cell wall is made up of two components. First, the water-insoluble material known as cellulose. Second, the outer pectic or mucilaginous substances. In addition to these, lipid, proteinaceous, and silicified materials are also present. Fourth criterion is the flagella. Flagella are present in all algae except red algae. Their number, their position, size, orientation, and whether they are smooth, what is called as whiplash, or whether they are hairy, what is called as tinsel, are important criterion for algal classification. At present, 9 algal groups are recognized, which are as follows: chlorophyta, euglenophyta, ctenophyta, chrysophyta, basidiophyta, heptophyta, cryptophyta, phaeophyta, and lastly the rhodophyta. If we start with the chlorophyta, it is commonly known as green algae. Green algae mostly occur in aquatic, freshwater or marine habitats, and many show symbiotic association. The photosynthetic pigments here are chlorophyll A, B, and carotenoids. They store starch as reserve food material inside plastids. The cell wall is composed of cellulose, hemicellulose, and pectic substances. And about flagella, they are either absent, or when they are present, their number is 2 or more. They are apical or subapical in position. They are equal or unequal in size, and mostly of the whiplash type. Now the second phyla is euglenophyta, commonly known as euglenoids. Euglenoids occur mostly in freshwater habitats and some in marine waters. The photosynthetic pigment here include, again, chlorophyll A, B, and carotenoids, thus resembling those present in the green algae. The reserve food material here is paramylon, a unique polysaccharide absorbed in the cytoplasm. Here the typical cell wall is absent. However, they have a flexible or rigid pellicle which is made up of proteinaceous strips just beneath the plasma membrane. Flagella, they are always present. They are 2 in number, subapical in position, and arise from a base of an anterior invagination called as reservoir. They are unequal in size. One is forward but an emergent one. The behind one is non-emergent. And they are of tinsel type. Next phyla is Ctenophyta, commonly known as dinoflagellates. Dinoflagellates mostly occur in marine habitats and some in fresh waters, and a few in symbiotic association, the famous one, the zooxanthellae. The photosynthetic pigments here include chlorophyll A, not B but C, together with carotenoids such as pyridinene, which is unique to this phylum. Typical cell wall is absent here and has a layer of vesicles beneath the plasma membrane with or without cellulose plates. The stiff cellulose plates look like a helmet forming a substance called as theca. Flagella are mostly absent and only present in the gamete phase. These are 2 in number, lateral in position, One is transverse, one longitudinal. They are unequal in size and they are of tinsel type. The next phyla is Chrysophyta, or commonly known as chrysophytes. Chrysophytes predominantly occur in fresh water and a very few in marine habitats. The photosynthetic pigments here include, again, chlorophylls A, C, and carotenoids such as fucoxanthin. The chief food reserve is the water-soluble polysaccharide called as chrysolaminarin, which gets stored in a vacuole found just near the posterior part of the cell. Typical cellulose wall is absent here, and the membrane is impregnated with silica scales or sometimes cellulose fibrils. The flagella are either absent or when present they are two in number, apical in position, equal or unequal in size. The forward one is tinsel and the behind one is whiplash. The next phyla is the Bacillariophyta, commonly known as diatoms. Diatoms occur in both marine and freshwater habitats. The photosynthetic pigments are chlorophylls A, C, together with fucoxanthin and xanthophylls. The chief food reserve include lipids and chrysolaminarin, which are stored in a vacuole. The cell wall is silicified. It's known as fristules. The cell wall consists of two halves which fit together just like a laboratory petri dish. Flagella are mostly absent and only present in the male gametes, and that too of centric type diatoms. Centric type diatoms show radial symmetry. Their number is 1, they are apical in position, and they are of tinsel type. Sexual reproduction is of spatial type here by oxospores. The next phyla is Haptophyta, commonly known as haptophyte algae. Majority of these occur in marine habitats and only a few in fresh waters. The photosynthetic pigment here are chlorophyll A, C, carotenoids, especially fucoxanthin. The food reserve is chrysolaminarin, stored in the vesicles in the posterior part of the cell. On the outer surface of the cell, scales of cellulose or calcified organic material is present. The calcified scales are known as coccoliths. Flagella, when they are present, they are two in number, apical in position, equal or unequal in size. They are of whiplash type. In addition to this, a thread-like structure called as haptonema, a characteristic feature, extends from the cell along with the two flagella. This haptonema differs from flagella both in its structure and its property, because they do not show typical 9+2 arrangement of microtubules, and haptonema can bend, coil, but cannot beat like a flagella. The next phyla is Cryptophyta, commonly known as cryptomonads. Cryptomonads occur in cold and subsurface waters of freshwater and marine habitats. The photosynthetic pigments here are chlorophyll A and C, also carotenoids, together with phycobilins. Cell wall is here absent but possesses a stiff layer of proteinaceous plates beneath the plasma membrane. The chief reserve food is starch. Flagella are present, two in number, subapical in position, and form the base of a depression they arise. They are unequal in size and tinsel type. Phaeophyta, next phyla, commonly known as brown algae. Brown algae almost entirely occur in the temperate and polar waters of marine ecosystems. The food reserve is laminarin, stored in the vacuoles. Cell wall is composed of cellulose, embedded in a matrix of mucilaginous algae in the inner wall layers. Flagella is present only in the reproductive cells. There are two in number, lateral in position, one tinsel forward and one whiplash behind. The next phyla is Rhodophyta, commonly known as red algae. Red algae predominantly occur in tropical marine habitats and a few in fresh waters. The photosynthetic pigments here are chlorophyll A and B and carotenoids together with phycobiolins. The cell wall composed of here is rigid inner component consisting of cellulose microfibrils and an outer mucilaginous layer consisting of sulfated polymer of galactose. Red algae are unique in the sense that they lack flagella. So with this we conclude this topic, wherein we discussed the general characters and classification of algae. Thank you.